Hello, everyone. My name is Edmundo Lozoya Gloria from Simvestab IPN in Irapuato, Guanajuato, Mexico. I would like to thank Dr. Peter Griffin for the invitation to this Congress on Plant Science and Biosecurity. I would like to talk to you about the work we have been doing in my lab since some time. That deals with metabolic responses of the microalga Neochloris oleoabundans to extracellular self and non-self DNA. This story started in 2006 with the Barton's group. That time they were working with the transmembrane proteins. Those are some receptors which recognize pathogen-associated molecular patterns, or PAMPs. Specifically, the TLR9 it's localized inside late endosomes or lysosomes. And with the work of Barton, they did a chimeric TLR9 receptor which now was localized on the surface, not into the endosomes, but on the surface of the cells. And in that location, the receptor was able to recognize self-DNA and trigger all the defensive responses after that. In 2015, Masoleni's group, they worked with plants in general. They realized that the extracellular DNA produces specific species inhibitory effects on plants. So after the plants in the soil die and they are degraded in the soil, and all the litter produced release some products, some chemical, biochemical, or physical. And among the biochemicals, they are the DNA of the diet plants. And if the plant recognizes the self-DNA, the reaction is very strong. In 2018, some colleagues from Simvestab Irapuato, they were working with the Phaseolus vulgaris plants, and they were interested to check the hydrogen peroxide production as an indication of the stress condition. They used DNA from Phaseolus vulgaris, from Phaseolus lunatus, and from Acacia farnesiana, and those DNAs were added to Phaseolus vulgaris plants. When this plant recognizes the self-DNA, the reaction was very strong. The amount of hydrogen peroxide increased in comparison with the DNA from Phaseolus lunatus and Acacia farnesiana. If the species or the DNA was isolated from similar or close species, the reaction was stronger, but not so high like with the self-DNA. Then in 2021, Cusiano's group were working with Arabidopsis thaliana to see the similar phenomena, and they extract the DNA from Arabidopsis thaliana as self-DNA and from herring as non-self-DNA. They added these DNAs to the plants, and as you can see in this panel, the self-DNA after 1 hour post-treatment, they was kept outside the cells, outside the roots, in comparison with the non-self-DNA, which was internalized and degraded intracellularly. Okay, the effects on the plants were also very dramatic. So the phenotypes after 10 days of post-treatment indicated that with the addition of self-DNA, the plants were very damaging. So they had inhibited growth, chlorotic leaves, and necrotic roots, in comparison with the plants were added, which were added with the non-self-DNA, which were really healthy plants. In microalgae, my group in 2016, we demonstrated that the microalgae also responds to stress conditions. We used at that time the colonial microalga Amautrococcus brownie, and as elicitors we added sodium chloride, sodium bicarbonate, acetic acid, and methyljasmonate. We checked the stress condition by the production of reactive oxygen species, or ROS, and we detected these ROS using the cell ROX dye, which in contact with these ROS molecules, they fluoresced, the dye fluoresced, indicating that the reaction was triggered. In the panel A or D, which is under microscope, we can see that the control, without any elicitor added, we couldn't see any fluorescence in comparison with the fluorescence obtained after addition of all the rest of the elicitors. We had very strong reactions after addition of methyljasmonate and sodium bicarbonate. So in this case, we demonstrated that the algae also reacts to elicitors with stress responses. In 2022, Palomba's group also worked with some algae like Lavinomola reinhardti, fresh water algae, and N. gaditiana from salt water. They added different concentrations of DNA, and they checked the reactions when they added non-self-DNA in the three concentrations, 3, 10, or 30 nanograms per microliter. The growing of the cells in both cases was the same, no problem. But when they added self-DNA, the growing of both cells, like in Chlamydomonas and HRT, was really affected. They didn't grow properly in both cases. So that was also an effect, dose dependent. In my group, we started to work with Neochloris oleoabundans, which is a microcellular algae, very attractive for the industry because of the amounts of lipids production, and it's a very easy algae to grow. So this is the growing curve of the algae, and after 10 days, we can get the maximum number of cells. And that time we collected the algae, extracted the DNA, and we used the DNA for the experiments. Those experiments were done after addition of several elicitors after 6 days of growing, when the exponential growth started to increase, and at that time we added the different elicitors. The DNA should be treated before addition because otherwise it doesn't work. This is an agarose gel showing the entire DNA extracted from the algae, and after sonication we fragmented the DNA and we obtained fragments between 200 and 700 base pairs with the self-DNA from Neochloris and with the non-self-DNA, and that time we used the salmon sperm DNA. We were interested to check the reaction of Neochloris after addition of these elicitors, and for that we assigned first the peroxidase enzyme activity as an indication of oxidative response. After addition of the self-DNA, as indicated by the green bars, as soon as 5 minutes after addition, the activity of this enzyme was the highest, and then the activity increased corresponding to different elicitors. For example, after addition of sodium carbonate, at 10 and 15 minutes the response was very high, but decreased after all this time. And with the methyl jasmonate, the reaction was very high at 120 minutes. But anyway, the reaction after addition of self-DNA was still high. We also checked some products derived from stress conditions like polyphenols, similar to plants. And we also checked that after addition of the self-DNA, as indicated here, after 48 hours, the amount of polyphenols was high. Not so like with addition of methyl jasmonate or sodium carbonate, but anyway, the amount of polyphenols indicated that the algae was really stressed and producing polyphenols to contrast these effects of oxidative metabolic response. We also assay the production of some phytohormones, which is also another indication of a stress condition. To avoid so many graphics, brief abstract of the results is that isopentyladenine, benzylamminopurine, and gibberellic acid, these three phytohormones were increased after 24 hours of addition of self-DNA. Methyl jasmonate and indole acetic acid didn't have any change along the time, 0, 24, or 48 hours, which were the times we assigned. But interestingly, the production of salicylic acid decreased after addition of all kinds of elicitors. At the beginning, it was in certain amount, and that amount decreased in 24 or 48 hours. That was an interesting response from the salicylic acid production. We also cited the lipids, because this algae is interesting because of the production of lipids. And after 24 hours or 48 hours after addition of self-DNA, this was the indication that the self-DNA triggered the maximal amount of lipids in comparison with the other elicitors. At 48 hours, the other elicitors, even the non-self-DNA, were producing some high amount of lipids, but nothing compared with the 24 hours. So the response was specific for self-DNA. The kind of lipids we were checking were the triglycerides, and this is an HPTLC imaging where you can see the standards, which were 3 different triglycerides of different length. And at 0 hours, 24 and 48 hours, all controls, non-treated cells produces the normal amount of triglycerides, so more or less the same in all times. But with the 3 different elicitors, methyl jasmonate, sodium carbonate, and mainly with self-DNA, the production of triglycerides is very, very high in comparison with the production of triglycerides in non-self-DNA addition. In all cases, the non-self-DNA samples were very, very low, and the samples with the elicitors, methyl jasmonate, sodium carbonate, and self-DNA were producing triglycerides in high amounts. Those results indicated a metabolic change, and we were also interested to check the amount of proteins produced in this change. So we checked the total proteins after addition of some elicitors or growing in different conditions. For example, it is quite well known that in microalgae, the condition of low nitrogen produces high amounts of lipids. We were checking also the amount of proteins, and at 0 hours, in all conditions or the addition of elicitors in control, low nitrogen condition, addition of self-DNA, combination of low nitrogen plus self-DNA in comparison with non-self-DNA. At 0 time, there were no changes, but at 24 hours, the condition of self-DNA added produced the biggest amount of proteins. Interestingly, we analyzed those proteins in acrylamide gels, in SDS-page gels, and as you can see, in comparison with the molecular markers we have here, the control non-treated cells produced specific patterns of bands of proteins, which are clearly seen here, and is the same pattern we obtained in the condition of low nitrogen with self-DNA or non-self-DNA. But when we added the self-DNA, the pattern of proteins changed dramatically, and we just indicated some of the more remarkable proteins. So we are now currently working on the proteomics of these samples to see if you can recognize some specifically proteins triggered by this addition of DNA. Up to now, we can say that microalgae responds as other living organisms to different DAMs, damage-associated molecular patterns, with the activation of ractide oxygen species, also the polyphenol content, and all this to alleviate the oxidative burst. Like in plants, we also were able to activate the phytohormone production. The response, in general, was more evident and stronger with self-DNA in all cases, in comparison with a very distant phylogenetic non-self-DNA like salmon sperm. So, as far as we know, this is the first evidence that self-DNA induces metabolic changes in microalgae. Finally, I would like to thank people involved in this work, mainly the students, Monica Zarate and Mariana Rojas, who were working directly with the algae, Claudia Rivera and Patricia Arroyo, which are current technicians helping us with the cultures of algae, Dr. Elizabeth Quintana and Dr. Doman Carorona from CIATEC, and we all together were guiding these guys for the work. And the funds came from different parts. I also want to thank to Texas A&M University, mainly biochemistry and biophysics department, where my colleague Timothy P. Divarin introduced me to work with microalgae, and Dr. Yvette Cornejo Corona, a former previous PhD in my laboratory student, and she also is working with Botryococcus brownie. We started this work with a call 2016 from Texas A&M University and CONACYT, and the rest of the funds came from the CONACYT, National Council of Science and Technology in Mexico. I thank you very much for your attention, and if there are some questions, I will be really happy to answer if I can. Thank you very much.